Hello and welcome back to Equity Tutors. Today we are going to continue on with the homeostasis module, and we are going to look at glucose regulation and specifically at the pancreas and how this is involved in glucose regulation. So like last lesson with Amelia, we'll first go over the structure of the pancreas, and then we'll go on to talk about how the pancreas has endocrine functions and what hormones are released and how they affect blood glucose levels. So the first thing I want to talk about today is the structure of the pancreas. So first of all, let's talk about where exactly the pancreas is. So the pancreas is an organ that sits under your liver and next to your spleen. So as you can see in the diagram, the pancreas is made up of three sections. So it's got a head, body and tail. So the pancreas has two functions. It has exocrine function and endocrine function. So the exocrine function is that the pancreas produces, stores and releases digestive enzymes which are necessary for our digestive system. And then the endocrine part of the pancreas is involved in secreting hormones that regulate metabolism and glucose. So what we're going to be focusing on today is the endocrine function of the pancreas. So the main parts of the pancreas that we need to know about with regards to the endocrine function are the islets of Langerhans. So as you can see in the diagram, there are these circular groups of cells which are called the islets of Langerhans, and then surrounding that are the acinar cells, which are what form the exocrine function in the pancreas. So you don't need to worry about those today. What we're going to concentrate on is what cells make up the islets of Langerhans. So the islets of Langerhans are made up of several types of cells, and they have different names and they secrete different hormones. So we're going to be focusing on the alpha and beta cells today, but there are also a couple of other cells within the islets of Langerhans, such as the delta cells, which secrete somatostatin, and the F cells, which secrete pancreatic polypeptide, but you don't need to worry about those cells too much. What we really need to concentrate on is the alpha cells and the beta cells. So the alpha cells secrete glucagon, and the way I like to remember that is that alpha has an A, so it secretes glucagon, so with the A in glucagon, and then beta cells secrete insulin. So obviously at this point we haven't spoken about what those hormones actually do, and that's what we're going to talk about in the rest of the lesson. But what I really need you to concentrate on to begin with is just being able to identify the structure of the islets of Langerhans. So you need to be able to, in your exam, to label the alpha and the beta cells and say which hormones they secrete. And you should also be able to draw a diagram with the acinus, so the acinar cells surrounding the islets of Langerhans. So they form a ring around the islet of Langerhans. You also need to remember that the islets of Langerhans also contain capillaries, and this is where the hormones released from the alpha and beta cells can enter into the bloodstream and be transported around the body. So as we learned about in the previous lessons, with hormones it's really important for them to reach their target tissues, so they need to enter into the bloodstream so that they can reach those target tissues throughout the body. Okay, so now that we've gone over the basic structure of the pancreas, let's go in to talk about the two main hormones that it releases, so insulin and glucagon. So we're first going to start off with insulin. So the effector cells that the insulin works on are the liver, muscles and fat cells. So they are the cells and the tissues that will respond to insulin that is released from the beta cells in the pancreas. So these are what we call the effectors. So these are what brings about the change that the insulin release wants to do. So the insulin is released, it affects these cells have a reaction to that, and that then allows the body to get back into its homeostatic range of glucose. So I've started this lesson saying that we're going to talk about glucose regulation, and I'm just now going to go into exactly why it's important to keep the level of glucose in our blood very constant. So as we know, glucose is used for energy. So if the concentration of glucose within the blood decreases below the level that we need it at, then the cells in our body won't have enough glucose to respire and to perform their normal function. So it's really important that the levels of glucose in the blood are maintained at a level in order for our whole body to function properly. Conversely, if the glucose gets too high in our blood, then it can disrupt the normal function of cells. It has a big effect on the water potential of our blood and things like that, and that can also cause major problems. So it's really important to keep the blood glucose within the right range. So that's a very important part of homeostasis within the body. So as I said, insulin is something that is used to modulate the glucose in our blood. So as I said, beta cells secrete insulin, and I'm now just going to talk about how that secretion happens, and then we will go on to the action of insulin within the target tissues. So the beta cells within the islets of Langerhans within the pancreas detect blood glucose levels, and when they increase too much, then the beta cells within the pancreas will start releasing insulin. So the important thing to remember here is when the blood glucose goes too high, insulin is released, which helps bring down the levels back into the normal range. So insulin is released when glucose levels are high, and conversely, glucagon is released when the glucose levels are low. So the way that this works is that glucose is absorbed into the beta cells of the pancreas, and this is done via facilitated diffusion via a carrier protein. So if you remember way back, we did go over facilitated diffusion and what carrier proteins are. So once the glucose is absorbed into the beta cell, the insulin containing vesicles within the beta cell move towards the cell surface, and they release the insulin into the capillaries. So this means that insulin is leaving the beta cells and going into the blood. So this works because when glucose goes up in the blood, the beta cells take more glucose in, and this causes a release of the insulin. So this again is a feedback loop. So you're getting the glucose is causing the release of the insulin. So then once the insulin is released from the beta cells, it goes into the bloodstream and circulates around the body, and then can act on its effector tissues, which, as I said earlier, are the liver, muscles, and the fat cells. And this stimulates the uptake of glucose by these cells to reduce the levels of glucose in the blood. I know that sounds like quite a lot, so I'm going to go over it again really quickly and just recap what I've said. So blood glucose is high. The beta cells within the islets of Langerhans detect that by taking in glucose through the carrier proteins via facilitated diffusion. This causes vesicles within these cells that contain insulin to move to the cell surface and release their insulin into the capillaries. So then the insulin enters into the bloodstream and can be transported around the body to have its effect on the liver, the fat cells, and the muscle cells. So once the insulin has gone into the bloodstream, this is now taking place outside of the pancreas. And now we're going to go into exactly what the insulin does and how this affects the blood sugar levels. So the same thing happens in all these different types of tissues, so in the muscle cells, the fat cells, and the liver cells, because they all contain this thing called a glucose transporter protein in their cell surface membrane. So that is actually the target receptor of the insulin. So All these tissues are slightly different, but the main principle of how the insulin works on them remains the same throughout. So what happens is that the insulin molecule will bind to the receptor on the cell surface, and this sends a signal within the cell to cause vesicles that contain glucose transporter proteins to the surface of the cell, to the cell surface membrane. So within the cells there are these vesicles which contain these receptors. The insulin binds to its receptor on the cell surface, and that causes a signal. Then the signal makes the vesicles move towards the cell surface, so more glucose transporter proteins are added to the surface of the cell. And this allows glucose to be taken into the cell, and this allows the glucose levels in the blood to reduce as the glucose is being taken out of the blood and into these cells. So the more insulin, the more receptors are put on the surface, and then the more glucose these cells can take up, and the faster they can reduce the blood glucose levels. So this is a general way that insulin works, but in the liver it can also do something else where it can increase the uptake of glucose into the liver by stimulating glycogenesis. So when a glucose molecule has entered into a liver cell, then an enzyme can convert it into glucose phosphate, and then this can be converted into glycogen by another enzyme called glucose phosphate. And this can then help to keep the glucose concentration within the liver cell lower to facilitate a steep diffusion gradient between the blood and the liver cells. So basically, the liver is able to convert glucose from the blood into a different form, into this thing called glycogen, which allows the concentration gradient of the glucose in the liver and in the blood to remain high, so that this process works well. So it's important to remember that it's only in the liver that this glycogenesis happens, and we're going to go into a bit more on this later on in the lesson. But the main thing to remember is that insulin causes a reduction in blood sugar by causing uptake of glucose into muscle cells, fat cells, and the liver. And specifically in liver cells, this glucose can be converted into glycogen. Okay, so now that we've spoken about insulin, we can now go on to talk about how glucagon works and how this is released from the alpha cells. So glucagon works in the opposite way to insulin. So glucagon is released when the blood sugar levels get too low. So we need, as we said earlier, we need to have enough glucose in our blood to allow respiration. So glucagon is there to make sure that that stays as high as it needs to be. So, as I said earlier, the alpha cells in the islets of Langerhans are what produces the glucagon. And what happens is that alpha and beta cells in the pancreas both act as receptors, and what will happen is if the blood glucose levels get too low, then the beta cells will stop releasing insulin and the alpha cells will start releasing glucagon. So the effector cells for glucagon are the liver cells. So remember, the insulin works on the liver cells and also on the fat and the muscle cells, but the glucagon only acts on the liver cells. So what happens is that if there is a decrease in blood glucose levels, then this is detected by both the alpha and the beta cells in the pancreas. And as I said, the alpha cells will start secreting glucagon and the beta cells will stop secreting insulin. So this causes the insulin concentration within the blood to decrease. So the liver and the muscle cells will stop using glucose. So they'll stop doing what we spoke about previously. So they'll stop taking the glucose into their cells. The way that glucagon works is in a way that we call the second messenger model. So this is because it has two main messengers within the system, and I'm going to go over exactly what happens. This is a bit more complicated than the insulin, so do take time to go over exactly what's going on here, because it will take a little while to actually get into your head what's happening at each step and the different names of the different things that are involved, especially the enzymes, because they do have very similar names. So do take time to go over this and make sure it's really in your head before moving on. So the first messenger is the glucagon, and this is released from the alpha cells, travels in the blood, and then it binds to receptors that are on the cell surface membrane of liver cells. So remember, this is only on the liver cells. So this binding causes a conformational change in the receptor, and that activates a G protein. So the activated G protein can then activate an enzyme called adenyl cyclase. So I know that sounds confusing, so let's just recap that again. So glucagon binds to a receptor on the cell surface, and once that's happened, it causes a activation of a G protein, which in turn causes the activation of the enzyme adenyl cyclase. You might also see this called adenylate cyclase, and either one is correct, so don't worry about that. So once this has bound to once the glucagon's bound to the glucagon receptor, it's activated the G protein. This has in turn activated the adenylate cyclase. Right. So the adenylate cyclase then can do its job, and this can then convert ATP to cyclic AMP or CAMP. So cyclic AMP. Now the cyclic AMP is what we call the second messenger. So the first messenger is the glucagon. This causes the activation of the G protein, and the G protein activates adenylate cyclase. Adenylate cyclase can then catalytically convert ATP into cyclic AMP. I know that is a lot, so don't worry if that sounds really confusing right now. You just need to go over it a lot of times and you will finally get it. If the only thing you remember from this is one thing, then please do remember that the second messenger is cyclic AMP. Okay, so now we're on to the second part of this cascade. So what happens is once you have this cyclic AMP, this then goes on to do a few other things within the cell. So the cyclic AMP binds to protein kinase A, which is an enzyme, and this activates the enzyme. So cyclic AMP activates protein kinase A enzymes. These enzymes then go on to activate phosphorylase kinase enzymes by adding on a phosphate group to them. So the phosphorylase kinase enzymes require an added phosphate group to become active, and this phosphate group is added by the active protein kinase A enzymes. Again, I know this is a lot, so really don't worry too much about it. It will become clearer over time. So once we now have this activated phosphorylase kinase enzyme, this then can activate glycogen phosphorylase enzymes. So we've got a whole lot of enzymes which act to activate other enzymes. So just try and remember each step as we go. They will become clearer over time, but kind of just get an idea of what is going on at each step. So even if you don't remember the names right now, just kind of think, okay, so the first part is one enzyme adding a phosphate group on to another one, and things like that. So get the general idea of what's going on rather than the specifics of the names. So we now have active glycogen phosphorylase enzymes, and these catalyze the breakdown of glycogen into glucose. So if you remember with the insulin, we could get glucose converted into glycogen. we can now do the opposite. So we can break down glycogen, which is a store that we can keep in our body for when we do have low glucose levels. And this enzyme breaks down that glycogen into glucose. And this is called glycogenolysis. So remember that anything with lysis in it means breaking down. So glycogen is the storage molecule, and glycogenolysis means breaking down of glycogen, and this releases glucose. So I know that is a hell of a lot that's going on, and we're going to go into a bit more on these processes in next week's lesson, so don't stress if it's not really sinking in now. We will recap it in a second, and then we will go into a bit more on things like glycogenolysis and things like that next week. So an important thing to remember is that this whole cascade that's happening with the second messenger, so the cyclic AMP, is an amplified effect. So basically one glucagon can activate this system, but then at each stage you're going to get more and more enzymes being involved, and it causes an amplified effect. So it causes the release of extra glucose by the liver, and this can bring up the blood glucose levels back to the normal range, which is what homeostasis is all about. It's returning the body to its normal range where the body works at its best. So I'm going to go over and recap exactly what is happening here at each stage, and then hopefully that will become a bit clearer to you. So first of all, alpha cells release glucagon into the blood. Glucagon is the first signal, or the first messenger. So the glucagon then binds to receptors on the cell's surface membranes of liver cells. So it binds to glucagon receptors that are on the surface of the cell. This causes a conformational change in the receptor. So a conformational change just means that it changes shape. So the glucagon binding to the glucagon receptor changes the shape of the receptor. And this then activates a G protein. So the G protein that is activated is then used to activate an enzyme which is called adenylate cyclase, or adenyl cyclase. Adenyl cyclase is then able to convert ATP into cyclic AMP. And now remember, cyclic AMP is now the second messenger. So now we're on to the second part of the cycle. So we've got cyclic AMP. Cyclic AMP binds to protein kinase A enzymes, and this causes them to become activated. The activated protein kinase A enzymes then can activate phosphorylase kinase enzymes by adding on a phosphate group. So the phosphorylase kinase enzymes are inactivated, and then they have a phosphate group added to them by the activated protein kinase A enzymes, and they become active themselves. So the phosphorylase kinase enzymes have this additional phosphate group to make them become active. These activated phosphorylase kinase enzymes then can activate glycogen phosphorylase enzymes, and these catalyze the breakdown of glycogen to glucose, and this process is called glycogenolysis. So glycogen and lysis, so that means the breakdown of glycogen. So this causes glucose to be produced, and this is then released into the blood to cause an increase in blood glucose levels to bring them back to the normal level. Okay, so I know that is very confusing and it will take a while to get it, but don't stress about it. The key things to remember here are: first messenger is glucagon. Glucagon binds to a receptor on the liver cells, and this causes a conformational change. This activates the G protein, which then causes the adenylate cyclase to be activated. ATP is converted to cyclic AMP by the adenylate cyclase, and then we have a series of 3 enzymes. So we have one enzyme is activated by CAMP. That enzyme then goes on to activate another enzyme into phosphorylase kinase, and then once the active phosphorylase kinase enzyme, that can then activate the final enzyme, which is the glycogen phosphorylase enzyme. And this is the enzyme, so this is the final one, so this is the one that does the conversion of the glycogen to the glucose, and remember this is the glycogen phosphorylase enzyme, so that is the one that actually helps to allow the process of glycogenolysis. So I know that is a hell of a lot, but don't worry, it will become clearer over time. I'm just going to now go over a quick recap of insulin and glycogen very briefly as a wrap up for the end of the lesson. Next week we will go on to talk about general principles of blood glucose regulation and the feedback loops involved, and then also talk about the problems when this system is not correct, so things that can happen once this system is not regulated properly. Okay, so let's begin by recapping what's going on in the pancreas. So the pancreas has endocrine and exocrine functions. The endocrine functions are performed by the islets of Langerhans, and these are made up of alpha and beta cells. Alpha cells secrete glucagon and beta cells secrete insulin. Insulin is used to reduce blood glucose levels, and it acts on liver cells, fat cells, and muscle cells. It causes these cells to take in glucose via glucose transporter proteins. Glucagon works in the opposite way. It is used to increase blood glucose levels, and it works through this 2-messenger system. So the glucagon binds to glucagon receptors that are expressed on liver cells, and that's important to remember that it is only the liver cells that express these receptors. And then this causes a whole cascade of enzymes, and the second messenger in this system is the cyclic AMP. So the first messenger is glucagon. This causes conformational changes in the receptor. It causes G protein activation and adenylate cyclase activation, which causes the conversion of ATP to cyclic AMP. Cyclic AMP then goes on to activate a bunch of different enzymes which activate each other, going down all the way to the bottom where we get the activation of the glycogen phosphorylase enzyme, which can convert glycogen into glucose, which then allows the glucose levels in the blood to increase to the normal levels. So as you can see, there is a lot going on in our bodies to keep our glucose levels correct. So we're going to go on to talk about that more next lesson, but just try and get all these processes in your head today. So especially what the insulin and the glucagon do and how they do it. That's really important to really understand before we move on.
Thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
Bye!